Hello, this is Staphylococcus Bacteriology tutorial presented by the SchrDbb. Reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So here we're going to visualize Staphylococci. So we need to know that among the classification of Staphylococci, we have free-living bacteria. So Staphylococci are inside free-living bacteria. Remember the classification upward in the introduction of bacteriology. We said that there were intracellular bacteria and there were free-living bacteria. Is it clear? And all of them were containing the cell membrane. Is it clear? So now among the free-living bacteria we contain the cell wall. So among the free-living bacteria is divided into two. We have the gram-positive bacteria and we have gram-negative bacteria. So Staphylococci are not among the gram-negative bacteria. So we're going to continue with gram-positive bacteria. Now among the gram-positive bacteria can be divided into cocci and rods depending on their shape. Is it clear? Now, among the cocci, we have Staphylococci, we have Endococci, we have Peptostreptococcus, and we have Streptococcus. All these are Streptococci. Is it clear? But there are different groups of Streptococci. This one is a group D Streptococci. This one I think group C Streptococci, and these are group A and B Streptococci. It's called Streptococcus. Now, among the Staphylococcus, we have Staphylococcus aureus, we have Staphylococcus epidermidis, and we have Staphylococcus saprophyticus. So these are the examples of Staphylococcus that we're going to visualize. Now in this stain, we can visualize a gram stain showing Staphylococci, just to tell you that the Staphylococci are gram-positive, and usually Staphylococcus aureus exists in cluster, as you can visualize here. So they are cocci who can exist in a group, like a bunch like that of grapes. Is it clear? Now let's visualize so we need to know that in Staphylococcus we have different things. So we have the Staphylococcus aureus. So we need to know that generally the significant host compromise is required for Staphylococcus aureus infection. So when you have a break in the particular mechanism barrier of Staphylococcus, that the Staphylococcus now can enter inside the body. Is it clear? So those are the different things that you should know. Now these are the causes of disease resulting from infection of Staphylococcus aureus. The first thing is that Staphylococcus aureus disease may be largely or wholly the result of an actual invasive infection. The first thing is that you may have an invasive infection that may result to Staphylococcus aureus invasion. So you start by colonization, then you have infection. Infection when you start by the contamination of the certain part, then colonization, then infection. Colonization is the growth of the microorganism, the division of microorganism, but without producing any signs and symptoms. While infection is when the signs and symptoms are being produced. So that's the first case of how Staphylococcus aureus can cause infection. The second thing is intoxication. So this is the Staphylococcus aureus itself that causes the disorder in the patient. In intoxication, Staphylococcus aureus disease may be largely or wholly the result of the toxin in absence of infection. So just saying that there is no infection that is due to Staphylococcus aureus, but is due to its toxins. So this is called pure toxicosis. Example in case of food poisoning. So in food poisoning, the Staphylococcus aureus is going to produce toxins, which are exotoxins, that are going to penetrate the GI walls, that is gastrointestinal walls, and then cause the patient to have gastrointestinal symptoms such as vomiting and abdominal pain. Now the next case where that can cause disease is in the case of both infection and toxins, where Staphylococcus aureus disease is as a result of combination of the infection and a combination of the toxin that is being produced. Is it clear? So when you have the combination of both like that, it can result now to still another disease. Now this so after visualizing how Staphylococcus aureus can cause the disease, we need to know certain virulence factors of Staphylococcus aureus. Why Staphylococcus aureus? Virulence factors are the different characteristic of a particular microorganism to cause a disease. So the virulence factors of Staphylococcus aureus, you have number 1, it contains a capsule. Number 2, so this capsule is a type 5 or a type 8 capsule. That's the first thing to know. Number 2, it contains a protein A, and protein A is a major component of Staphylococcus aureus cell wall. Is it clear? And it binds to the Fc portion of the immunoglobulins, existing an anti-opsonine effect, that is an anti-phagocytic effect. The next one, factor that can reduce that can increase the virulence factor of Staphylococcus aureus based on the cell wall characteristic is the fibronectin binding protein. And the fibronectin binding protein and other Staphylococcus aureus surface protein promote the binding of the mucosal cells. So it's capable of binding to the mucosal cells just to invade those cells. Is it clear? We have the next one, the clumping factor. The clumping factor, this is a fibronectin binding protein that enhances the clumping and the organism. Is it clear? So it enhances the clumping of the organism in presence of plasma. Now the next, after knowing these are the cell wall virulence factor. Is it clear? After knowing the cell wall virulence factor, we have other factors of Staphylococcus aureus such as the cytolytic exotoxin factor. So you have it produces exotoxins. So the exotoxin that are produced, we have the alpha, the beta, the gamma, and the delta toxin that affect the mammalian cells, including the red blood cell membrane, that can cause hemolysis. Is it clear? So that's this next thing. Now we have the Panton-Valentine leukocidin. The Panton-Valentine leukocidin, this is a pore forming toxin disease of the polymononuclear neutrophils. Is it clear? So you have mononuclear neutrophils. So it's just for neutrophils. So we continue with the fact that we have the next, which is in the virulence factor, we have seen the Panton-Valentine leukocidin, and we said that this is a pore-forming toxin that is going to cause lysis of the polynuclear cells, and that polynuclear cells are usually neutrophils. Is it clear? So that when they kill those neutrophils, it's now going to result to the invasions of the Staphylococcus aureus. Now it is mostly present with methicillin resistant Staphylococcus aureus. Is it clear? Methicillin resistant Staphylococcus aureus strains that we're going to visualize later. Now the next virulence factor is the super antigen exotoxin. With the super antigen exotoxin, these toxins have an affinity for the T cell receptor major histocompatibility complex class 2. Is it clear? So they are capable of simulate the enhance of T cell T lymphocyte response, and many as 20% of the T cell response compared to the 0.1% that respond to usual process antigen. So you're going to make the T cell to respond. Is it clear? Now for the super antigen exotoxins, now we have the super antigen exotoxins is going to be divided into these different toxins. The first toxins, you have the enterotoxins. The second toxin is the toxic shock syndrome toxin, and the next is exfoliating toxins. Actually, these super antigen toxins make sure that since it stimulates the major histocompatibility complex class 2, is going to make sure that the T cells are going to identify the normal cells of the body as being invading cells. Is it clear? So when it identifies the normal cells of the body as being abnormal antigenic cells, it is going to destroy the normal cells of the body. Is it clear? So these exotoxins here are produced by the Staphylococcus aureus. They are going to bind on the receptors of the normal body cells, and then when they bind on the receptors of the normal body cells, the T cells are going to bind now on those receptors, and then visualize that there is going to be an abnormal. So there is going to be now immune response on the body based on those exotoxins. The first is the enterotoxin. The enterotoxin consists of the 6 major antigenic types. We have A, B, C, D, E, and G. So I produce approximately half of all the Staphylococcus aureus that we isolate are going to produce the enterotoxins. When these bacteria contaminate food and are allowed to grow, this secret enterotoxin, and the injection which can cause food poisoning. Is it clear? So the toxin is going to stimulate vomiting center of the brain by binding the neuroreceptors at the upper gastrointestinal tract. Is it clear? So that's how the enterotoxin works. The next toxin that is going to be produced are the toxic shock syndrome toxin. This is going to be classic. This is the classic cause of toxic shock syndrome because it is the same molecule that is going to be associated with the toxic shock syndrome, and then it is referred to the enterotoxin F. You see that in this ladder there was A, B, C, D, E, and G. There was no F. So the enterotoxin F is considered as the toxic shock syndrome toxin because it's mostly associated with the disease called toxic shock syndrome that we are going to see later. Now we have exfoliating toxin. It's also a super antigen. It is caused by it is going to cause now what is called scalded skin syndrome in children. That's the exfoliating toxin. Is it clear? So now let's visualize now the super antigen exotoxins now summary. So you need to know that the toxin have an affinity to the T cell receptor major histocompatibility complex class 2, as we have already said initially, and we need to know that the toxins stimulate the enhancement of the T cell response. Since it's going to enhance the T cell response, you see that the immune response is going to be very great. Is it clear? So the T cell activation now can cause now a toxic shock, because you need to know that in T cell activation you have the release of the interleukins, you have the release of the interferons, and all these release now are pyrogens, and they are also going to be release of prostaglandins. Is it clear? So the patient is going to have toxic shock. When we say that the patient is toxic, it means that the patient has all the symptoms of like fever, like palpitation, like increased frequency of respiration, tachypnea, and all that. So these are the symptoms of toxic. So toxic symptoms are symptoms when there is excessive stimulation of the sympathetic nervous system. So that's when the patient is said to be toxic. Now shock occurs when as a result of hypotension. Is it clear? When the blood pressure of the patient is less than 90/60 millimeters of mercury, resulting to hypoperfusion of the different body organ, the patient is now going to have what is called shock. Is it clear? So it's going to be by the release of large amount of the C cell cytokines. So the super antigen exotoxin, after causing all these particular characteristics like enterotoxin affecting the GI tract, the toxic shock syndrome affecting the skin, causing and the exfoliating also causing scalded skin syndrome. After causing that, the super antigen may also result to a toxic effect and a shock effect. Is it clear? Particularly seen in the toxic shock syndrome. Is it clear? So that is what is seen. So you have antigen present, you have the T cells. Is it clear? So you have T cells, you have antigen presenting cells, and you have the super antigen. The super antigen come at that major histocompatibility complex that is going to be found between the antigen presenting cell and the T cell, and is going to increase the immune response. So in this patient, you're going to try to reduce the immune response of the patient, else the patient is going to have excessive immune response that may even kill the patient. Is it clear? So now the next one is in this diagram now you can see other virulence factors of the Staphylococcus, like the enzymes. So enzymes are other virulence factors of Staphylococcus aureus. Now Staphylococcus can also produce different enzymes like Protease enzyme, Catalase enzyme, the hyaluronidase enzyme, and the fibrinolysin enzyme. Is it clear? So all that is going to be produced. The next is the slime production, particularly Staphylococcus epidermidis. Staphylococcus epidermidis produces slime. The next one is the cytolytic exotoxins. You have already seen the different exotoxins that can affect the mammalian cell, but these cytolytic exotoxins are not the same like the super antigen exotoxins. Is it clear? For the cytolytic exotoxins are just exotoxins that are going to result to hemolysis, are going to result to lysis of the cells, while super antigenic exotoxin cause an increase in antigenic property. Is it clear? Cytolytic exotoxin only results to the lysis of the cell, just like the red blood cell is going to cause hemolysis, and they are referred to as hemolysins. Now, the next, we have also a protein A binding. The protein A is another factor. Okay, protein A is another virulence factor that is going to bind at the level of the FC receptor. Is it clear? So when that protein A binds to the FC moiety of the immunoglobulin gene, and is going to exert an anti-phagocytic effect. Normally, the immunoglobulins is going to cause opsonization, that is going to favor the binding of the phagocytes to the Staphylococcus aureus. But this protein A now is going to prevent that opsonization, anti-opsonin effect. The next virulence factor is going to be fibronectin, which is shown here, and the fibronectin binding protein is located there. Is going to bind to fibronectin that promotes the binding of the mucosal cells. Is it clear? So normally it knows to know that if you want to adhere to a cell, you must find a certain receptor on that cell for you to adhere on that mucosal cell, and you have the fibronectin binding protein that can be helping to adhere on the fibronectin which is present on the normal cells of the body. So all these are the virulence factors. Is it clear? Now the next thing are the disease caused by Staphylococcus aureus. Now we start by the skin and the soft tissue infection. So the skin and soft tissue infection, you need to know that you have furuncles, you have carbuncles, you have wound infection, just like the traumatic and the surgical wound infection. You have cellulitis and you have impetigo. Is it clear? So those are examples of skin and soft tissue infection caused by Staphylococcus aureus. Now respiration infection caused by Staphylococcus aureus, you have pneumonia. Muscular infection caused by Staphylococcus aureus, you have osteomyelitis. Musculoskeletal, you have arthritis. Cardiovascular infection, you have endocarditis, you have septicemia. Genitourinary infection, you have renal carbuncles, and you have lower urinary tract infections. Now these are disease caused by the toxins now of Staphylococcus aureus. You have toxic shock syndrome, you have scalded skin syndrome, and we have food poisoning like gastroenteritis. Is it clear? So these are the different disease caused by Staphylococcus aureus. Now the next one, now we have, so these are the different cases where you can see toxic shock syndrome with the different explanations. We have Staphylococcus gastroenteritis with different explanation, and we have the scalded skin syndrome producing the bullae on the skin with different explanations. So now you can just pause and then visualize the different characteristics of the disease, and you can also pause here to visualize different characteristics of the disease here. I'm not going to speak more about that, but you need to know that in toxic shock syndrome is characterized by the different the peculiar characteristics that you have fever, you have hypotension, you have multi-organ dysfunction. The multiple organ dysfunction is just due to the shock. Is it clear? So, and then the toxic is because the patient is going to have high sympathetic nervous system stimulation, just like fever. The patient is going to have fever. The patient is going to have tachypnea, is going to have tachycardia. Is it clear? Multiple organ dysfunction, and you have an erythematous rash with a desquamation occurring due to convalescence. Is it clear? So that's another one. So that is the desquamation that you can visualize. Now in scalded skin syndrome, it involves the appearance of superficial bullae that is going to result to exfoliative toxin that attack the intercellular adhesive stratum granulosum. Is it clear? So usually we need to know that what the scalded skin syndrome, it just resembles like a bullous impetigo. Impetigo, you just have small vesicle to push on all through the body. Is it clear? But now bullous impetigo, you also have bullae, just like scalded skin syndrome. But the difference is that in scalded skin syndrome, just like the word implies, is that the skin is going to peel off due to the toxins, while in bullous impetigo, usually the skin do not peel off and they contain pus. Now, these are the different. So, this is one method how you can recognize Staphylococcus aureus. You need to know that there were different enzymes that were contained inside Staphylococcus aureus. Like example, a catalase positive culture of Staphylococcus aureus. Is it clear? So, the test for catalase is performed by removing a colony to a slide of drop of 3% hydrogen peroxide. You see now, you place catalase on hydrogen peroxide. You place on a normal culture media, and then you place hydrogen peroxide. You see that the cells are going to grow, and then a certain part you are going to have but air bubbles that are going to be produced in the culture media. Is it clear? That's for due to the production of oxygen, because they produce catalase that can break down hydrogen peroxide to produce water and oxygen to produce the air bubbles. Now, what you need to know is the next thing you need to know is the treatment of the Staphylococcus aureus. So you need to know the choice of the antibiotic is going to be difficult now because with Staphylococcus aureus, because the Staphylococcus aureus is now resistant to many antibiotics. Is it clear? It's resistant to penicillin G, and you also have penicillase encoding plasmids that can produce the penicillase. Is it clear? So that's why it's difficult. It's difficult now to produce. But usually, what you have to know is that Staphylococcus aureus is resistant to the number of beta-lactam antibiotics such as methicillin, oxacillin, amoxicillin. Now they are now resistant to all that. You have hospital-acquired Staphylococcus aureus, which are methicillin resistant Staphylococcus aureus, and you also have community-acquired methicillin resistant Staphylococcus aureus. Is it clear? So in hospital-acquired methicillin resistant Staphylococcus aureus, you have different mutations that may result. I'm not going to speak about those mutations. You can just visualize it up here. Now, what are the comparison between hospital-acquired methicillin resistant Staphylococcus aureus and community-acquired methicillin resistant Staphylococcus aureus? Is it clear? The PVL is the Pantone Valentine leukocidin. So that Pantone Valentine leukocidin was mostly focused on the virulence factor that was found at the level of methicillin resistant Staphylococcus aureus. So let's visualize the characteristics of the patient. For hospital-acquired methicillin resistant, the patient are typically elderly, debilitated, and chronically ill. While for community-acquired methicillin resistant Staphylococcus aureus, the patient is typically young, healthy, children, athletes, and even military. Is it clear? Now, the infection site. Bacteremia commonly occur with no obvious infection site in hospital-acquired methicillin resistant Staphylococcus aureus, and the infection is usually surgical wound, open ulcers, intravenous line, and urinary catheter often occur. Now, in community-acquired methicillin resistant Staphylococcus aureus, the infection is usually on the skin, on the soft tissue, producing cellulitis abscess. The infection also includes necrotizing community-acquired pneumonia, and you have septic shock syndrome. Is it clear? Now, the transmission of hospital-acquired methicillin resistant Staphylococcus aureus. So the transmission occurs within a healthcare setting usually, while the transmission is in a community here. For the medical history, infection more likely in patients with the history of methicillin resistant Staphylococcus aureus infection. Is it clear? Like recent surgeries, admission in the hospital, and nursing care, while the patient show no significant medical history of the infection in community acquired. Now, virulence of the infecting strain. Usually, the spread of the infection in the community is limited, and the Pantone Valentine leukocidin gene is usually absent. So you see, the virulence is not going to be very high in hospital-acquired methicillin resistant Staphylococcus aureus, but the virulence, like in the virulence factor of the Pantone Valentine leukocidin gene, is going to be more present in this case here. Now, the antibiotic susceptibility. Here you have multi-drug antibiotic resistance often occur, and resulting in limited choice of effective therapeutic agent. Here, the strain are often more virulent than hospital-acquired methicillin resistant Staphylococcus aureus, but they are susceptible to broader antibiotic array than in hospital-acquired methicillin resistant Staphylococcus aureus. basically. Now the next chapter that I'm going to visualize are the coagulase negative Staphylococcus aureus. Example of coagulase negative Staphylococci, not Staphylococcus aureus. Example of coagulase negative Staphylococci, we have Staphylococcus epidermidis, Staphylococcus saprophyticus, basically. So those are some examples of coagulase negative. So you can visualize a diagram here showing Staphylococcus epidermidis that's going to be attached to a biofilm and going on the surface of a catheter. So now here is going to be a summary of the various species of the Staphylococci. And now we have Staphylococcus aureus, we have epidermidis, and we have Staphylococcus. The frequency of the disease caused by aureus is going to be common, epidermidis is going to be common, and Staphylococcus is going to be occasional. Now you need to know that it is only Staphylococcus aureus which is coagulase positive, meaning that if you place it in a blood film, it's going to clot the blood, basically in presence of plasma. Now for epidermidis is coagulase negative, Saprophyticus is coagulase negative. The color of the colonies is clear. The color of the colonies of Staphylococcus is golden yellow. Color of colonies of the Staphylococcus epidermidis is whitish, and Saprophyticus is variable. Mannitol fermentation. In case of fermentation, you need to know that it is aureus that is mostly involved with fermentation of mannitol. Epidermidis no, and then that is no. Novobiocin resistant. So aureus is not resistant to that. Epidermidis is not resistant, and then Saprophyticus is resistant to novobiocin. Now this is a summary of Staphylococcal disease. 1 is going to indicate the first line drug, and 2 is going to be indicating the second line drugs. So the Staphylococcus species, we start with the Staphylococcus aureus. Now we need to know that the Staphylococcus aureus can cause skin and soft tissue infection, as you have already seen: osteomyelitis, septic arthritis, endocarditis, septicemia, necrotizing pneumonia. So we have necrotizing pneumonia, we have toxic shock syndrome, and we have food poisoning. So antibiotic therapy is not used in case of food poisoning. So when you have food poisoning, it is not because of an infection that the patient is going to have the disease. So it is mostly because of the toxins. So in food poisoning, no antibiotic is used there. So now, Staphylococcus species, when you have Staphylococcus aureus, what are the different antibiotics you use? Provided that it is a methicillin susceptible strain, you can use oxacillin, you can use nafcillin. If it is a methicillin susceptible strain, is it clear? Now, if it is a methicillin resistant strain and it is obtained in healthcare associated, it means that it is a healthcare patient that is going to have that methicillin resistant Staphylococcus aureus, or it is any patient that has been in contact with the hospital and is having a methicillin resistant Staphylococcus aureus, you mostly give vancomycin. Now for methicillin resistant, but it is mostly but in this case community acquired, is it clear? So with a mild to moderate infection, you give now a thimetoprim sulfamethoxazole, is it clear? Or you give doxycycline. So those are the different things you give. Now in case of a methicillin resistant with community acquired disease, and then it is severe infection, in this case it is mild to moderate, you give sulfamethoxazole, thimetoprim, and doxycycline. But if it is community acquired and then severe, see with methicillin resistant, you give daptomycin, or you give linezolid, or you give vancomycin, or you give teicoplanin. Teicoplanin is an example of other penicillins, teicoplanin, or you give quinapristine, daltopristine. Is it clear? So these are the different treatment that you use here. So now, in this diagram, you can visualize the Staphylococcus aureus that is going to be cultured from a wound, and you see how they are gram positive with clusters. Is it clear? Now, Staphylococcus aureus on blood agar surrounded by the zones of beta hemolysis. You need to know that on the blood, you need to know that there were certain toxins that were produced, that cytolytic exotoxins produced by Staphylococcus aureus, that's going to cause hemolysis. Is it clear? And then you see the colonies of the Staphylococcus is going to be more yellowish in color. Now, the characteristic of Staphylococcus aureus is said that it is catalase positive, it is non-motile, do not form spore, they are round cocci tending to occur in bunches like grapes, they are facultative anaerobic organisms. Is it clear? So Staphylococcus aureus are more anaerobic than aerobic, but facultative means that they can be both anaerobic and they can also occur as aerobic organisms. That's why they place the word facultative. The facultative, anaerobic, or even aerobic. Is it clear? So we have cultured on enriched media containing broth and even blood. Is it clear? So the next now is Staphylococcus epidermidis. So Staphylococcus epidermidis is usually the infection of catheters and heart valves is usually due to Staphylococcus epidermidis, and usually there are methicillin susceptible strains. You can use oxacillin, you can use nafcillin. Now, in case where you have a methicillin resistant strain, that you can use now vancomycin. Is it clear? So most isolate resistant to penicillin G used in methicillin resistant isolate. Now for Staphylococcus saprophyticus is going to mostly cause cystitis in women. So what you use there is ciprofloxacin. So now we need to know these are different diagrams of infections. This is a carbon core that is going to be caused by Staphylococcus aureus. This is how it resembles. It's going to be localized here. We have a furuncle caused by Staphylococcus aureus. The next is folliculitis, infection of the hair follicles caused by Staphylococcus aureus. You have the scalded skin syndrome caused by Staphylococcus scalded skin syndrome, and therefore you have superficial impetigo. Is it clear? So from here we are finished with all the videos involved with Staphylococcus aureus, and say thanks for your kind attention.